Buenos días a todos. Tenemos hoy el placer de tener con nosotros a un científico muy querido por todos y muy reconocido internacionalmente, que ya es parte de la familia de acá de la Escuela de Medicina, el doctor Mariano Garcia-Blanco. Mariano es puertorriqueño. Es actualmente catedrático y director del Departamento de Bioquímica y Biología Molecular en la Universidad de Texas, en Galveston. Tiene la silla distinguida Mildred Hyatt Bassett y John Román Bassett en su universidad. Es también profesor de enfermedades infecciosas emergentes en la Escuela de Medicina de Duke en Singapur. Mariano hizo su bachillerato en la Universidad de Harvard y su MD y PhD en biofísica molecular y bioquímica en la Universidad de Yale. Y continuó con un postdoctoral en biología de RNA con Philip Char. Después estuvo en Duke University hasta unos pocos años atrás, que pasó como director de departamento en Texas University en Galveston. Y hoy nos va a hablar de la helicasa de RNA DDX39B en español. Es un guardián de tolerancia inmune. Con ustedes, el doctor García-Blanco.
Bueno, gracias Adelfa y gracias a todos por venir. Ahora, antes de que asesine el lenguaje castellano de mi padre y de mi madre, voy a cambiar el inglés para durante la charla, pero siempre es un gran placer estar aquí en la universidad. Hace 2 años creo que no he pasado por acá, y la última vez que vine hablé de la otra parte del laboratorio que trabaja en virus. Hoy voy a hablar de la parte de que trabaja en cosas más celulares, pero que todo tiene que estar relacionado, desgraciadamente, porque yo no soy inmunólogo, pero tengo que aprenderlo. Estamos yendo a la inmunología. So today I'm going to tell you about this half of the lab over here. This is interesting. But the other side of the lab is what I talked to last time I was here, and we work on viruses that are RNA viruses, particularly flaviviruses like dengue, Zika, and yellow fever. Next time I come in 2 years or so, I will talk to you about what we're doing on this side of the lab. I have to put this up. I am forced by the university to tell you guys that I have some conflicts. In particular, related to what I'm going to tell you about today, I have started a small company in Texas, which actually is commercializing some of the early studies that I'll mention today. So that is a conflict, and I have to acknowledge it. I'm not worried about it, but it's a conflict. So today, I don't know if I'm going to cover everything. We'll go through the first 1/3 is published. The last 2/3 is all unpublished, hopefully to be published soon, but we don't know. So we'll talk about multiple sclerosis and autoimmunity. We'll talk about the interleukin receptor, interleukin 7 receptor, which is what led us to this whole story. And then the discovery that this protein DDX39B, which has many other names, and I'll tell you the other names, that immunologists discovered it first, then RNA biologists rediscovered it, and then we put it together, the two parts of, and then I will, at the end, have some speculative idea of how RNA binding proteins play extremely important roles in immunity. But let's start with multiple sclerosis, a disease that has been recognized for over 100 years. It's one of many autoimmune disorders. I don't have time to tell you an enormous amount about it, but it is essentially a demyelination disease in the central nervous system, and it's caused by multiple different components of the immune system, including cells in the central nervous system, T cells in the periphery, B cells in the periphery. Almost every aspect of the immune systems are involved in multiple sclerosis and other autoimmune disorders. Today, I'm going to focus more on T cells, as is the focus of our work. There are really 4 clinical classifications of multiple sclerosis, and most of what I'm going to tell you today has to do with the most common type, although what I'm going to tell you about probably relates to other types of multiple sclerosis. So most of what very formally, what I'm going to tell you applies to what's called relapsing remitting multiple sclerosis, which is the most common type. As you probably know, multiple sclerosis is a complex disorder, like many other autoimmune disorders. There are environmental components, but there are also very strong genetic susceptibilities, and we're going to focus on the genetic susceptibilities. That is, it's not a Mendelian disease like cystic fibrosis. That is, if you have, and this is something I want to make very clear when we go through the allelic variants that I'm going to talk about. These are variations that if you go around the room, everybody will have different alleles. It's not determining that you will get multiple sclerosis if you have the risk alleles. It just means that you have a higher susceptibility to get it, okay? And probably the most important thing about this is this pedigree down here. And it's to highlight that in many cases, but not all cases, many times autoimmune disorders travel together. And this is a pedigree from actually a classmate from medical school of mine. This is him down here. Thankfully, he's normal, but his mother had lupus, and his uncle died of multiple sclerosis, and he has a cousin with multiple sclerosis. And, you know, doctors love to say anecdotal things, but what I can tell you is that through the years, we've actually gathered many, many pedigrees that suggest that this is actually a pretty typical pedigree for families where there's autoimmune disorders traveling through the family. So the belief is that maybe there are shared genetic susceptibilities that then, depending on what stimulus excites the immune system, you either develop one type of autoimmunity or another. That's a general idea of why that may explain this. Now, not every case of autoimmunity or autoimmune disorders travels in families. There are people who have MS that have never had a relative with MS, right? So that happens also. So we're going to talk about what was the first genetic loci that was shown to be associated with multiple sclerosis outside of the major histocompatibility class 1 or 2 regions. And this is work of a good friend and colleague. There he is, Simon Gregory at Duke University. And what Simon and his team of human geneticists did is they actually identified the first SNP, or polymorphism, and it was in this gene, the interleukin-7 receptor gene. And as you can probably tell by looking down here, this is the SNP that they found in this genome-wide association studies, and it was found in a small exon of the interleukin-7 receptor gene. And I'm not a human geneticist, and at that time, this was in the mid-2000s, I was not working on autoimmunity. But Simon came to me and said, look what I found. And I said, so why should I care? Other than the fact that, I always liked autoimmunity as a general concept. And then he said, well, it's in the small exon in the gene. And very quickly, we went into Ensemble, and I said, not only is it a small exon, actually, I said it's actually not that small. The exon has 94 nucleotides, which is not that far from the average exon in our genes. But I said to him, the beauty, I just found out just by looking at the browser, it is an alternatively spliced exon. So then I became very interested. And in fact, the gene makes a lot of sense for it to be related to autoimmunity. I'm not going to go through everything that's in there, but it's absolutely required for homeostasis and the correct behavior of T cells. So what we discovered and worked together and published it in a couple of papers that are now quite old was that the way that the allele actually, our prediction is the way that the allele mediated multiple sclerosis is the following. First, a disclaimer. The allele is a non-synonymous allele. So people who have the C allele have a different amino acid than people in the receptor, than people who have the T allele. Unfortunately, Simon had a postdoc that spent 2 years, and all that she could show was that the signaling through those two variants was exactly the same. She could find no difference in the signaling. Now, you can never prove a negative, right? So there may be some contribution of that, but the major contribution is what happens to splicing. If you have the C allele, which is the risk allele, you actually splice 3 times more frequently going down this way than if you have the T allele. Okay, the numbers are still dramatically in favor of making this. So what happens? Oh, this is a new one. Great. Oh, I like this one better. So exon 6 is where that SNP is, okay? And exon 6 encodes the only transmembrane domain of the protein. So when you include exon 6, the receptor goes to the membrane and everything's okay. And this is the majority product in both cases. It's about 90% if you're homozygous T. It's about 70% if you're homozygous C. So not a big change. Now this is a big difference. This goes about 2 to 3-fold higher if you're homozygous C. And this is a soluble protein. All right, so our work suggested, based on genetics and molecular biology, that this is the reason that the SNP is active. That you get susceptibility when you make more of this. And then Lauren McCall at Stanford published a very nice paper where she artificially introduces soluble IL-7R into mice and then exacerbated an MS-like syndrome that you can induce in mice called EAE. Okay? Now, one thing that's important to remember for the rest of the talk is that this alternative splicing, this is a classic case of alternative splicing where you have a cassette exon that either gets included or gets skipped. This particular splicing event does not happen in any other organisms than primates. So this is a late evolutionary development. Only primates make this soluble form. So this experiment, although completely artificial, injecting it into the mouse, gave some functional backing to our idea. This also presents a problem for us because the mouse is not a good model to study this reaction. But that was a discovery that started this whole project. And obviously, let's go back here for a minute. Whoops, I'm pressing all the wrong buttons here. This, given that we thought that the susceptibility to develop MS had to do with the decision to either splice or not splice this exon, we decided that we wanted to know everything there was to know that we could find out about this exon. So first a student, Irina Evskova in the lab, and then a postdoc, Gadiel Galarza, who's from Puerto Rico, actually a graduate from the Neurobiology Institute, figured out how this exon works. And that's what we're going to tell you this part of the talk, which is what regulates IL-7R exon 6. This is Gadiel. He's looking even scarier now. He has a huge, long beard now. And Gadiel started the company with me, so he's still in Texas. But Gadiel decided to say, okay, what are the proteins that regulate this exon? And together with work that Irina had started in the lab, they actually said, okay, one of the ways in which you can do this, you know what they say? They say, in the past, they used to say, if you didn't understand something, you did genetics. And now what I say is, if you don't understand something, you do omics. you first see what sticks to it, right? So what he did was, what proteins stick to this RNA? So he took the RNA from the exon, and he found a bunch of different proteins, and 12 of them were actually really good candidates because they actually bound only when certain elements were there that we knew were important for regulation. And then he took each of those 12 and he functionally tested it. And only one of those proteins gave us the expected change in splicing. And you'll see it in the next slide. And it was this protein, DDX39B. I have to admit to you, when he first showed me the data that said DDX39B, I had no idea what it was. And I'll tell you why in a minute. Because this is a new Hugo name. So yes, DDX39B. And here's some data. This has been published already, so I'm not going to belabor it. But essentially, if you knock it down, and you cannot knock it out, we cannot knock this protein. It's essential. So the cells will die if we don't have any of it. So we can get it down to like 30 to 50% of the levels in a control. And you can see there is the knockdown. And when you do that, what you see is you have now a lot of skipping. So this protein is required for the exon to be included. So what happens when you do more skipping, and this is a reporter, this is endogenous, these are cell lines, this is in primary human T cells. So you will do more skipping, and then you make more soluble IL-7R in the supernate. Okay, so we became convinced that functionally this protein did what we were interested in. It was a very important activator of the exon. But then this is a little bit of why I didn't know what this was. The way I knew the protein has UAP56. That was the second name that the protein had. This was the first name, and it was discovered by immunologists. It was called B-associated transcript 1. Jack Strominger discovered it back in the 1980s. And then also, it was also shown in the same year that Gadiel published his paper. A group published it as being important in an androgen receptor splicing event. And I have to say that there is a paralog of this protein called DDX39A. And a lot of the early work on that people call UAP56, it's not really clear which of the two paralogs they were talking about. Okay. So then we decided, okay, we have all the molecular biology, genetics, that says this protein is important biochemically for this reaction. Can we connect it back to autoimmunity in some ways? And what we did was we collaborated with two human geneticists that had actually also collaborated with us in the early Nature Genetics paper. And we were not completely truthful with them. Okay, this was a single blind experiment. We told them we had purified 122 proteins that we were interested in. We didn't tell them anything about our functional data. And we said, we want you to go back to, they have the largest collection, they're part of the largest consortium for human genetic variation in multiple sclerosis. So we said, go back to your tens of thousands of patients versus tens of thousands of controls and tell us any of these proteins, the genes that encode them, do they have any association? And they found one. And obviously we celebrated in the lab that day because it was DDX39B. So now we had biochemistry, molecular genetics, and now we had human genetics supporting that this was important. And in fact, I never thought I was going to publish a paper that had a Punnett square because I'm not a geneticist, but this was impressive. If you are, so this is now stratifying populations of individuals. These are not patients and controls, stratifying for risk to develop MS. And here are the alleles in the original IL-7 receptor. These are the alleles in DDX39B. If you are homozygous for the risk allele for both, and you may note something curious here, the risk allele for DDX39B, which is A in our nomenclature, is very, very rare. The risk allele for IL-7R is not, and we can talk about that after the talk. It is overrepresented in MS patients, but it's not a rare allele. But if you're homozygous for both of those, and you compare yourself to a homozygous for the protective alleles, you have almost 3 times higher chance of developing multiple sclerosis. So that was a very impressive effect. In fact, this effect is almost as large as the effect that is conveyed by the haplotype in the major class 1 of the HLA genes, essentially. So that was nice. I'm going to go very quickly through this, but to tell you that we now understand what we think we understand. I should always be careful of that, but we believe we understand how the DDX39B allele works. And it is an allele in the 5' UTR of the gene. So it's not part of the protein coding sequence. It's an allele in the 5' untranslated region, and it affects the level of the protein. So in people who are at risk, so this is the rare AA, this is an African population, this is an Iberian population of lymphoblastoid cells. So this is in vitro genome-wide association study. We can see that the level of the protein is very low among the AA. And we were able, Gadiel was able to show using molecular reporters that that SNP could alter the translational efficiency of the messenger RNA. So explaining molecularly what the basis of the SNP was. So then this is the picture as it was towards the end of 2017. We had 2 alleles in the human populations. One controls the level of the transacting factor, DDX39B, which the transacting factor pushes the equilibrium this way. So when you have low levels of DDX39B, you go that way, you go down. As they like to say in the US, you go south. But now I live in Texas, so that's kind of insulting. And then in something that actually I have a very, very smart student right now who's figured out now what the molecular reason for this is. It's not quite ready for prime time, but it has to do with the assembly of a complex that actually DDX39B acts on that complex and de-represses the exon. So that paper should be finished this year, I hope. And but the important thing is that the combination of these 2 alleles gives you a susceptibility to go one way or the other. And that was really, really nice. And we felt like we had gone from human genetics to molecular explanation. And let me just say that since we've done this work, very similar things have been shown for other diseases, particularly type 1 diabetes. So we believe that this mechanism is not only active for multiple sclerosis, but actually is probably important in other autoimmune disorders. But then we had a quandary, and that this is now Gadiel looking more corporate now that he's the president of the company. And although all of this work, he was still a postdoc. And then he was joined by, and I apologize, I actually misspelled Minato's name. I had one of the typical, what do you call that, ADD or maybe dyslexic moment, which I didn't think I had, but his name is actually Minato Hirano, and I just noticed that I made that. So I inverted his, so Minato is a really, really smart postdoc. He worked on West Nile virus before and tick encephalitis. He was a flavivirologist from Japan. He's a veterinarian and a PhD. Came to the lab, gung-ho to work on dengue, and then saw that autoimmunity work and switched. He went into what we call the dark side of the lab. So, and what we were asking is, you know, I told you that splicing reaction is only conserved in primates. But DDX39B has been conserved through every vertebrate, and actually its location is conserved, and I'll tell you towards the end, in every vertebrate. So there was an idea, this protein's probably doing other things. And the immunologist, way back when, had actually said that this protein was a transcription factor that was anti-inflammatory. I think they were 50% right. It's not a transcription factor, but it's definitely anti-inflammatory, or at least it worked that way in our first example. So we decided, okay, is it doing anything else in autoimmunity? And I wouldn't ask a question here if I didn't have an answer that was pleasing. So it's a little, so what we decided to do, once again, when you don't know anything about something, you do omens, right? And so we took CD4 T cells from 2 patients. Actually, we did it from 6, but we didn't do 6 RNA sequencing libraries. We only did, we did 6 patients, and we took 2 of them, and we all the 6 we treated the same way. We transduced it either with a control shRNA, a lentivirus, or with 2 independent shRNAs knocking down DDX39B. And so these are donor 1, and you can see there's enormous variability between donors. And so this represents here the most stringent type of analysis to looking at what RNA transcripts are dysregulated when you knock down DDX39B. And because this 134 here in the middle represent those that are affected by, checking my time here, by both independent shRNAs in both donors. Okay, so we decided to focus on these 134 transcripts that were altered. And we immediately found what we call the shining pebble. And I have to admit, I'm not an immunologist, so I said, oh, the first time I read this, the name of this protein, I said, transcription factor. I don't want to work on transcription. Wow. And then, you know what? You go through PubMed, and if I would have been an immunologist, I would have jumped immediately up and down. When I checked this in PubMed, at that time, there were already over 9,000 references for this particular protein. Right now, there's over 11,000 references. That was only a year and a half ago or 2 years ago. It's ridiculous. So this protein is very well known. It is a member of a family of transcription factors, but it is absolutely essential for the development and function of T regulatory cells. When I was taking immunology in graduate school, they were called suppressor T cells. So that's how things have changed. It is also absolutely very well known to be linked with autoimmunity. FOXP3 is the cause in mice of scurvy and in humans of IPEX. These are devastating autoimmune syndromes. If you don't have, these are null alleles of FOXP3. If you have mutations in FOXP3, you will have a massive autoimmune disorder because you don't have T regulatory cells, which dampen down autoimmunity. And in human studies, and this is kind of an embarrassing point, it has been genetically associated with multiple sclerosis and with type 1 diabetes, but the associations have been relatively weak. And it has been weak because people haven't tried hard enough, because FOXP3 is in the X chromosome. And the majority, 90% of the GWAS that have been analyzed have excluded the X chromosome. That is an embarrassment. Why? Because it's harder. It's harder to use genes that are on the X for obvious reasons, for gene dosage. But it's amazing that nowadays they're, so we've gone back. When I first asked my colleagues about their imputations of all the other genes, they said, oh no, we never looked at FOXP3 because it's in the X. And I said, MS is a disease that happens 3 to 4 times more frequently in women than men. What's the deal here? But so that's changing now. But it is, I couldn't believe that. But that's because it is difficult, right? But still, it should have been done. So I told you we had done this in 6 volunteers. So we went back and then checked to see, can we, instead of doing the RNA sequencing, it's kind of expensive. We actually did sequence 2 of the other donors. And the sequencing was very deep. By the way, we did 350 million reads per sample because we want to look at very rare alternative splicing events. Although what I'm showing you here is all transcript level data. And yet we could confirm, again, with some variation, this is at the level of the RNA, this is at the level of the proteins, that when we knocked down DDX39B, FOXP3 went down, the RNA, and the protein went down too. And then we isolated T regulatory cells from several human donors and see the same thing in Treg. So FOXP3 is primarily expressed in Tregs. So we thought all of the CD4 T, so which is a combination of different types of T cells, all of that data came from the Tregs in that population, probably. But then we repeated the experiment again using actual purified, well, they're not pure. I'll show you, I think I have here a single cell sequencing experiment. They're far from pure. These cells really are a mixture of different populations, but they're enriched for Tregs. And very clearly, if we knock down DDX39B, we actually get lower levels of FOXP3, both at the level of RNA and the level of protein. And it's actually interesting that given the variation between donors, sometimes we have trouble with statistics for the protein we're knocking down, but the FOXP3 is knocked down as well. So this is one of the rare cases where we've seen a downstream target affected as much as the one we're actually targeting itself. So we then did a biochemical experiment, and for this we use a Treg-like cell line. It's an MT2. It's not a real Treg. It's a Treg-like cell line, and showed that if we divided if we fractionated into cytoplasm, nucleoplasm, and chromatin, the bottom, the take-home message is right here, that you can see the decrease in FOXP3 RNA, even in chromatin. You can see it in nucleoplasm, suggesting that whatever the DDX39B is doing, it's doing it very early in the biogenesis of the FOXP3 message. So I'm going to make it a long story short to tell you that we detected weak but compelling evidence that there was something called intron retention. So we could see higher levels of intron reads in some of these tracts of RNA sequencing. And very quickly, we then checked both in T cells and in cell lines that yes, the introns were being overrepresented. So it's as if the transcript was being made when DDX39B was low. We still had a lot of FOXP3 transcript, but it wasn't getting spliced. That the introns were being retained. And that eventually, we now know, leads to instability of the message, inability to make protein, instability of the message. And then Minato did he made a very important observation. And that is, he noticed that the FOXP3 gene has really weird introns. At least 9 of the 11 introns are really different. If you take all the X chromosome genes and you actually do pictograms of what the splice sites would look like, so this is an average frequency at a particular place, and here is a 3' splice site. That's the so-called invariant AG dinucleotide. That's the end of every intron, and then it starts with that G, and G is the beginning of the exon. And then you look upstream, it's something that we and others from the early 90s, we had called polypyrimidine tracts. And there really are poly-U tracts. And I apologize, this should be U's, not T's, because this is supposedly representing RNA. But if you actually looked at the FOXP3 introns, what it was a tract of C's. And in fact, FOXP3 represents an extreme. If you now take all the protein coding genes in the X chromosome and ask, what is the nucleotide frequency in this area of every intron in those genes, the majority of them are exactly where you expect them. They mostly have U's in there. These are the ones that have, this is the frequency of C's, and here is where FOXP3 is. It's that sort of pink dot up there. So it is an extreme version, although not the most extreme. And more recently, Minato has taken a couple of these genes up here, and they behave exactly like FOXP3. They're absolutely sensitive to this helicase, and they don't splice well when you knock down the helicase. So the interesting thing is that this is not just something that happens in humans. This C-rich pyrimidine tracts are found in FOXP3 genes all the way to platypus. So in every mammal, in all classes of mammals, all types of mammals, we were able to see that the FOXP3 gene has actually inherited these strange introns, suggesting that this is not an accident of evolution, but rather that the FOXP3 gene has been built, has been built to be regulated in that way, to be essentially transcribed at relatively good levels, but then be sort of at the edge of being able to be spliced in a way that really depends on the levels of this DDX39B helicase. And so this was all very pleasant to us, and because back in 1993, we had published a paper saying that C's were bad in the polypyrimidine tract. So this took us way back to my second student, Robert Rosigno, and Mike Weiner was a medical student. And we actually said, C's are not equivalent to U's, and we shouldn't be calling the polypeptide tract polypeptide tract. It should be poly-U tract, except that I had discovered a protein that had been named polypeptide tract binding protein, so we were stuck with a name. We were done. But the bottom line is that this was not a big surprise, that these C's were actually not good. So Minato very quickly went on to show that is a case, that yes, these, if you actually make reporters, you can actually put introns in the middle of a luciferase open reading frame and say, how sensitive are they to knocking down DDX39B? And now these are siRNAs that knock DDX39B. And you can see that the reporters that have the introns from FOXP3 are very, very sensitive to low levels of the helicase, whereas a control intron from human beta globin here really doesn't care. In fact, one of the repeats goes up, but we only believe things when we see it happening with both independent SIs or SHs. So suggesting that yes, these introns are bad, and then making mutations in those introns, Minato went on to show that it was truly the polypeptide tract that made the difference. So here is intron 11 of FOXP3, and we have the same exact data for intron 7. And then Minato makes a change to make the 5' splice site even better in that intro. It's better, but it's still sensitive to the knockdown of the helicase. Whereas if we convert the very C-rich tract in this intron into a poly-T tract, now it becomes insensitive to the knockdown of the helicase, suggesting that it is that step that really is important. So this is not that important, but actually what we see is that this is actually conserved in other FOX genes. The reason to show this is that we then looked at FOXP1, which is a gene that has very much the same structure as FOXP3, very, very similar structure, except it has more introns. But it has normal introns, and not surprisingly, when Minato tested those, they're really not in any significant way affected by knocking down the helicase. So it really is, within the FOXP3, it is the helicase that's really, it's the polypeptide tract that's determining the sensitivity to this helicase. And once again, when he looks at now, it's a different way of looking at this. Before, remember, we looked at what was the distribution of the polypeptide tract frequencies in the X chromosome genes. And he saw that FOXP3 was an outlier, way, way too rich in C. This is a different analysis. Now he actually saying, if I take 800 introns that are affected by DDX39B versus 800 random introns, how do they look like in the polypeptide tract? And again, what we can see here is that primarily the biggest difference that you see, both in terms of statistical significance, but also of changes, is that the introns that are affected by DDX39B actually have a reduction in U frequency and an increase in C frequency. There are other changes, but nothing is as dramatic as those two. That explains why these genes are so sensitive to this helicase. I'm not saying that the other genes are not in any way affected by the helicase. These genes are the ones that are most sensitive to the levels of the helicase. And remember, if we go back to the human genetics, you have variations that go about 2 to 3-fold in the level of the helicase among humans. Okay, so I'm going to skip through this part just to tell you that we've sort of put this in the context of a model that this is affecting. I'll just use this to summarize it. This UAP56 DDX39B is affecting how you actually form these early splicing complexes, and we have data to suggest that it is really at this step of the coming of the so-called U2 SNRP into the very early pre-spliceosome that is being caused by the changes in level of the helicase. And these introns are very, very poor at taking this first step in the splicing reaction, and that they require much more DDX39B to do it than many others. And I don't have time to go through all the biochemistry, but we've shown now that what they require is the ATPase activity of the helicase, and they really don't care about the helicase activity, which is a big surprise. But again, suggesting that it's a very specific step, because this step here just requires the ATPase activity of the helicase. So I'm going to go through that relatively quickly and get back to some more immunology. So So we now have figured out in pretty broad terms what the mechanism is for FOXP3 regulation. So you have low levels of DDX39B, that leads to low levels of FOXP3. So what are the consequences of that? Okay. Well, not surprising, this is a GSEA analysis saying that if you actually look at FOXP3 targets, they're affected, right? So this is essentially saying that if you look now, this is distributing published FOXP3 targets, and these are genes that are affected. And FOXP3 is both an activator and a repressor of transcription, but the majority of the genes that are FOXP3 targets we see are overrepresented in this part of the scale, which is the control, in other words, where DDX39B is high. So when DDX39B is high, FOXP3 targets are high. Not a big surprise. But it was nice to see this. And in fact, when we look at markers of Tregs, both FOXP3 and CD25, we see very clear changes. When we knock down DDX39B, we lose both FOXP3, as we have seen before. We also lose CD25, another marker of Tregs. Okay, so we looked at several other important immune regulators that Tregs produce, like IL-10, and this is now in primary human Tregs from human donors. There's a very steep decrease in IL-10 production after DDX39B knockdown. And when we actually looked at transcription factors, and this was a very telling thing, these are transcription factors that actually mark different lineages of T helper cells. So these cells are losing, what I showed you here is they're losing their Treg characteristics. So we knocked out DDX39B, not surprisingly, you lose down FOXP3, and you lose a lot of the downstream Treg characteristics. So they're not Tregs anymore, or at least they're not behaving like Tregs. But they're now starting to behave like cells that are either confused or are Treg-like. And here is, you have different transcription factors, TBX21, GATA3, ROR3, and this is in these cell lines, and the same has been seen in Tregs. They start acting as if they were T helper cells. They're not, in fact, they're somewhat confused, and I think I have it here. These are, when we start measuring now in the secretion, what these Tregs secrete, what we see is that they have some of the characteristics of Th2 cells, but they're not perfect Th2 cells. They don't do everything that these helper Th2 cells do. But they're secreting IL-5, they're secreting IL-13. These are from two donors, two Tregs. So these cells are starting to move away from being Tregs and perhaps being towards a T helper-like phenotype. And this is our one single cell sequencing experiment. So we took these supposedly Tregs. As you can see, they're quite complex in their in the clusters of cells that are there. These right here, this cluster here, and then this cluster over here too, are the two most conventional Tregs by markers. So what you do is you take, whoa, what was that? So you take these Tregs, you knock down, you have a knockdown control, knockdown of the DDX39B, and then you do sequencing, and we sequence about 2,500 cells for each of these two conditions. The cells are then in a essentially, this is an imaginary two-dimensional map based on the expression clusters of the cells. And what we see is that the clusters that are more like Tregs are diminishing. And then what this experiment told us, particularly this cluster 5 up here, this cluster 5 are cells that show actually primitive markers. So these cells now, when DDX39B goes down, you now have these cells that are essentially confused. They don't know exactly which lineage they belong into. And one concern about this is that those cells could actually be quite autoimmune. So not only are you reducing the level of the T regulatory cells, you're making these cells now more likely to be activating against self-antigens. So we believe that this RNA binding protein is critically important to mediate the splicing of FOXP3. FOXP3 determines Tregs. And it is known from this work, but also from previous work where people have actually dialed down FOXP3 in mice, that even with small decreases in FOXP3 levels, you start getting the T regulatory cell activity to go down, and you get this conversion. Depending on the tissue and the model, you get Th2 or Th17-like cells forming from the Tregs. But in any case, that's been presumed to be very dangerous because these T regulatory cells have T cell receptors that recognize self-antigens. So if they become helper cells, that will then induce autoimmunity. So the idea is that this is protecting from autoimmunity by maintaining high levels of FOXP3 and therefore high levels of T regulatory cells. And when you have low levels of DDX39B, these introns, there's only 2 in FOXP3 that splice fine, but 9 of them don't splice. You end up with an immature messenger, pre-messenger RNA that doesn't give you protein. So that's how we believe it controls it. So we put this together now, and this is why we call this a guardian of tolerance. And there's another third story I don't have time to go into, but on the one side, what we actually published in 2017, it affects this ratio, pushing the equilibrium up, away from this autoimmune mediator soluble IL-7R. On the other side of the equation here, we have, it pushes FOXP3 splicing here, increasing the levels of FOXP3, which increases levels of Tregs that then lead to autoimmunity. So we put this protein towards the center of autoimmunity and immune mechanisms. And what I'm going to try to tell you at the end, and this is very, very speculative, is that we believe that this is part of a whole program that connects RNA biology to immunity. And the reason we believe that is that Jack Strominger and others who discovered a lot of these proteins in the class 3 region, they were sort of baffled because some of them, like complement and TNF, they were obvious candidates for why there would be immune genes, right? So you have class 1 and the class 2 region, and then in the middle, you have this class 3 region, which people are finding a little confusing because some of those genes make a lot of sense. They look like immune genes. But a lot of the other genes, like this BAT1, as he called it, they had no idea what it was. But more recently, what people have, and people have published this based on 2 hybrid and interaction, but they had never really actually made sense out of this. A third of the class 3 proteins are RNA binding proteins. And this is conserved through all vertebrates. And it's really not clear. So as I was telling Adelpha, this is going to be I think maybe the last chapter of my career, is figuring out why are all these RNA binding proteins conserved in the class 3 region? Why is it that that evolution has maintained that? I don't want to anthropomorphize it, but it is, I'm very much of an evolutionary in the way I think. If it's there, it means they're doing something. And there is this connection, this ancient connection between RNA and immunity, because if you believe, like I do, in the early RNA world, the first genomes were all RNA. So the first infection was one genome trying to get into the other. And the first immunity was an RNA mechanism preventing the other RNA getting, get out of my house. You're not my RNA, right? So this idea that we think about DNA genomes, but in the beginning, as I say, it was RNA. So I think that this may be a fossil of that time. Some proteins like this one here is fascinating. We've known it for many, many years, that it actually is involved in decapping messenger RNAs. But nobody knows what it does. And the same thing, these are other exosome components of processing machineries, and nobody really understands it. So we're proposing to actually have driven by what we've done with DDX39B, trying to understand what these proteins are doing. I thank you for your attention. I'm sorry I may have gone a little long. I want to give credit to the people who did the work. For many years, I did this together with Shelton. He's now left on his own. And then Minato and Gadiel really were the two principal individuals in the laboratory, and then some amazing collaborators, Simon, Dennis at Duke, Georgia, and several, Farron and Lisa, they're all wonderful collaborators. And obviously I want to thank the funding, without which none of this could have been done, and all the volunteers who donated their sample, particularly patients with MS, who have been amazing, always super generous, donating samples for our work. Thank you very much.